Right, thanks for the nice introduction and for giving me the opportunity to speak here today about aspect analytics and what we do to support spatial multi-omics data integration. Today, I'll give a brief introduction of who we are, what we do, and then I'll dive into our approach for spatial multi-omics and make it tangible with some case studies, one of which will be the same project that was presented earlier by Professor Voet on the human prostate project, but we'll approach it from a slightly different angle. And then there's a few more case studies, and I'll end with an overall conclusion. Very briefly, Aspect Analytics, we are a spatial biology bioinformatics solutions provider. So we provide both software, which is a cloud-based platform called Weave that allows you to manage, visualize, and analyze your spatial biology data, and can be extended either by us for you or by your own bioinformatics team via an SDK that we provide, which I'll give a little bit more information of later as well. On top of the product, we provide hands-on bioinformatics services as well, where we take data provided from any collaborator and work on new developments. Typically, this can range from high-end new multi-omic integrations across different assays, but we also focus a lot of work on bringing individual assays to high throughput, where the bioinformatics challenges are somewhat different in the sense that it's very focused on QC and making sure that if we reach a certain stage of analysis for a sample, that a lot of assumptions have been validated to be met. So this is also work that we do quite a lot. In general, what we try to do and support the field is making spatial multi-omics easier, more accessible to everyone. And I'll show in a variety of use cases how we approach this in practice. I've listed some of our collaborators below, and you can see that it ranges from big pharma companies, power users like CROs, all the way up to leading academic institutes. So we span the whole range of fundamental research up to fully applied high throughput pipelines in practice. I don't think I have to explain the utility of spatial multi-omics to anyone here, but essentially it's about bringing together two key factors: spatial context as well as a multi-omics profile of our biology to understand everything that's going on. We all know this is relevant and useful, but it comes with a couple of challenges, because usually if we want to obtain multi-omic readouts, we're typically limited to having to use a variety of different assays. If I want to do transcriptomics, proteomics, and say metabolomics, I'm going to be using 2 or 3 different devices, typically on serial sections. And we have to somehow consolidate all this different data that is acquired in different formats, different spatial resolutions, and everything. What I also want to highlight here is that spatial biology, by its nature, is a very interdisciplinary field, and a lot of the challenges that we see and try to overcome using both our software tools and our services is collaboration between stakeholders from a completely different background. So we have wet lab experts, biologists, pathologists, bioinformaticians, and we all have to come together to tackle these complex data analysis problems. I won't talk too much about what we do in this area today. I'll focus on the multi-omics integration, but if you're interested in this, please come talk to us at our booth as well. In general, at Aspect, we have very wide experience across different assays. Most of the commercially available ones we have experience with, and not just analyzing data from that one assay, but integrating with all its friends that are on this slide. So when we talk about integrations, it's between all these different assays, and we have live demos of this available. In general, approaching spatial multi-omics starts from what we call a stack of serial sections obtained from a single sample, where individual sections are analyzed using different instruments. If you can run multiple assays on the same section without interference, please do. It makes life easier. But in general, many of these assays simply cannot be combined. So we end up with a disjoint set of measurements acquired on serial sections using different instruments. If we want to integrate this, we have to first consolidate to a shared coordinate system across this whole stack of measurements, which can range anywhere from 1 or 2 sections up to 10 or even more, depending on what people are trying to do in an analysis. And we have to consolidate all these different data formats from the various instruments, etc. Within our software platform, we have a process called Stack Fusion that turns this set of measurements into one integrated data frame. Think of an AND-data object or a pandas object, where for every observation, we now have the readouts from the various assays consolidated as different features. And this enables what we call true spatial multi-omics in the sense that you're not limited to being able to analyze whatever a single assay can measure at the same time together, like transcriptomics and a couple of proteins, but you can mix and match any combination of assays to target whatever you might want. Of course, this kind of data structure is not the end of an analysis, it's the very beginning. You can now, from a bioinformatics perspective, start from this integrated data structure and do whatever analysis you might want downstream. I have more examples of this coming up. But we can also do multimodal visualization, as we can see on this slide. So here, this is a spatial proteomics data set acquired using a Luna-4 Comet assay. On the serial section, we have a multi-imaging data set. This is spatial lipidomics. You can also appreciate a completely different spatial resolution, but we can visualize all of these together, fully interactive, whether it's one sample or several. All of these things are available. And it's not just visualization, it's also actual analysis, of course. All of this is made possible through our cloud platform called Weave, which is one centralized hosted solution where your various spatial biology data sets and even non-spatial things like single cell data or bulk proteomics can be uploaded, centrally managed, analyzed, and visualized together. For projects running in large consortia, this also enables collaboration because you have one centralized system where everyone can remotely access the same resources, either through the web UI, the only thing you need there is a browser, or via a set of APIs that we maintain for bioinformaticians to leverage the platform in their own scripts. Lastly, what I think is interesting to know is that we are the only software solution in spatial biology that's ISO certified for information security management. So we take data confidentiality very serious, given the applications that we work with. So your data is safe with us. Now let's dive into some case studies, and I'll start with the project that Professor Voet already indicated about prostate cancer. So part of the introduction, I think, is already done very well, so I don't have to repeat this. The main thing here is we started from 16 samples coming from 8 patients. 8 samples were malignant and 8 were non-malignant. And essentially what we combined is traditional histology and several spatial transcriptomics assays, in this case, Visium, not the Visium HD, the regular one, and spatial lipidomics, which in this case was done using an Orbitrap instrument. As explained in the previous talk, there was also single nuclei RNA-seq data available that was used in various ways. I won't go into detail on this. I'll focus on some other aspects. As discussed in the previous slide as well, we started from a stack of 4 tissue sections here per sample. One was used for regular histology, one for multi-imaging, one for spatial transcriptomics, and one for single nuclei RNA-seq. The first step here is to go to a common coordinate system, which is done through a series of image registrations, where we have to co-register across sections, which we did via H&E images that were available for every section. If that's not the case, we can also work with via other modalities, but in general, having a shared modality to do co-registrations across sections makes life a lot easier. Whether it's H&E, simple tissue autofluorescence, or anything else, it helps. What we also had to do, of course, is co-register the individual spatial biology assays, in this case, mass spec imaging and Visium, to their corresponding H&E. For Visium, this is implicit. For mass spec imaging, this is a process we have to do manual. The main thing that is important about any co-registration here, even if it's on the same section, is that they're always non-rigid. So you can have complex tissue deformation, especially when working on serial sections, but even when running individual complex assays on the same section, your tissue can swell and deform in various ways that we have to be able to account for in registration. This only solves one part of the challenge, which is putting everything into a shared coordinate system. After registration, we can move freely from one section to the other, one assay to the other. But as also indicated in the previous talk, there is still the difference in spatial resolution that we have to take into account. So what you can see here is that the circles, which are the Visium spots, have a completely different spatial footprint than the mass spec imaging pixels here, shown in squares. The spots have a diameter of 55 micron, the pixels in this project of 30. So when we want to go to a shared list of observations, we have to decide what do we want as an observation. In this particular project, what we did is we integrated everything to the level of the Visium spots, the circles you see on this image. And we did that by applying a Gaussian weighting, where essentially we made an aggregate mass spectrum based on the spatial overlap of every spectrum with a single Visium spot. And at this point, we have an actual integrated data structure where we now have, for every Visium spot, the whole transcriptome information that the Visium assay gives you, plus a representative lipid spectrum from the mass spec imaging, which was acquired on a serial section. If you would have run more assays, say a spatial proteomics assay on a serial section, we can keep using the same idea to add more features to this integrated data structure. And then we can do downstream analysis. In contrast to the previous talk, I'll focus mainly on spatial analysis here, particularly on correlation. And what we can find, starting from these integrated data structures, is gene-lipid pairs that are spatially co-expressed or anti-expressed in one sample, in several. We can stratify however we want. And it's not just pairwise like you see here, but we get a full correlation matrix between all genes and all lipids that were available. And here we're only showing the ones that have a very high positive or negative correlation. But this is a trivial analysis once you have the integrated data structure. But we can go further than this as well into what we call differential correlation. And in this case, what we did is we looked for gene-lipid pairs. And here on the left, we see a lipid. On the right, we have a gene. That are spatially correlated in normal samples, which you can appreciate if we look at corresponding samples, the spatial distribution is very comparable, but the spatial correlation is lost in malignant biopsies. And this is interesting because we have completely different omics layers, and we see that there is a mechanistic change induced by the disease, which we would not have seen without having access to the multi-omics or having access to the spatial data. So this is quite a nice application. This, of course, will feed into future projects in terms of trying to tease out what this actually means. A second case study I want to talk about is something we did together with MD Anderson and the University of Texas about ovarian cancer, where the setup was quite similar in the sense that we started, in this case, we were looking for to better understand the occurrence or reoccurrence of high-grade serous ovarian cancer. The default treatment is a combination of surgery, resections, and chemotherapy, but a lot of patients relapse over time, and it's unknown why or what the markers might be. And this is characterized by so-called minimal residual disease, which at MD Anderson, they try to identify 6 to 8 weeks via laparoscopy after the primary treatment. In this case, we had a cohort of 8 samples, 4 MRD positive, 4 MRD negatives, and the stack consisted of a first section where we did the 10x Visium assay, a serial section, the Luna-4 Comet, and then another serial, multiple flavors of mass spec imaging, in this case for metabolites, glycans, and peptides, which are all analytes that are unavailable to most other assays. So here we have a somewhat more complex stack, but we apply the same methodology, being combining modalities. We use the immunofluorescence primarily to bring us known information about cell types and so on based on their protein markers. But now per cell type, we can find metabolic profiles, glycan profiles, peptide profiles via the mass spec. So the first step is, again, registrations, where we first aligned the COMET data with the H&E of the mass spec imaging data, and then the mass spec imaging data as well, to get, again, a shared coordinate system. The next step, again, is the same idea. We have to define what is our unit of observation, which in this case, we define to be the mass spec imaging pixels, which, as you can see in the figure in the bottom, they're slightly larger than individual cells. So every pixel covers multiple cells. And what we have in the integrated data structure here is per pixel, the cell type proportion, the original antibody markers as well, but now peptide, metabolite, and glycan information, as well as transcriptomics, which I won't go into detail here because that analysis is not yet fully finalized. We can do the same thing, correlations between any pairs of these metabolites, but we can or different types of analytes, but we can also do different style data analysis. For instance, a differential expression analysis of metabolites or glycans or peptides between cell types. In this case, for instance, tumor or cancer-associated fibroblasts or any combination we might want, where we are leveraging both the proteomics data and the mass spec in this case. A third analysis that we did in this particular project is exploring the tumor stroma interface, where this was identified using proteomic markers. And then we're looking at, do we see changes in cell type density or individual marker expressions, both from the proteomics experiment, but also from the glycans, metabolites, peptide signals as a function of where we are in this interface to better understand the biology. There was one more example, but I will skip this in the interest of time. We did a reproducibility analysis between xenium assays. If you're interested in that, you can also talk to us at our booth. But in conclusion, we provide a cloud platform for spatial multi-omics with a rigorous standardized way to integrate across assays, to integrate across omics layers acquired on serial sections. And we've applied this to many different use cases with good results. Next to the software, we also provide services where we do hands-on data analysis and help you prototype new types of applications. And in general, Aspect is here to address the challenges in spatial multi-omics and bringing spatial multi-omics into high throughput. That's it.